thank you all for coming out tonight to the Queens Library here at Sunnyside. We've got a fun program for everybody. We're going to be growing edible mushrooms, or starting some edible mushrooms to grow, showing everybody how that's done. I want to stress right here at the beginning that I am not a mycologist. I work for the library. I'm kind of a jack of many trades, specialist of none. I did put on this workshop a few weeks ago at the Steinway Library for the first time, and we did things a little bit differently. We're doing a different method for sterilizing our growing medium tonight. So it's kind of a new workshop all over again. So I'm happy to be teaching it, and I'm happy that I have so many here in the crowd. Shirley has a sign-up list. If you just walked in or you haven't signed up yet, please do. That's the email list for Green Queens Library newsletter, and you'll find out about more events like this. We have environmental screenings of documentary films. We have garden shift opportunities for people to come and volunteer. We have compost collections at 4 different libraries. We're going to be expanding to 6 libraries in the near future. And we have other workshops like this. I'm going to be teaching people how to do hydroponic window farms next month. And a lucky patron will be taking that system home as well whenever we do that. So please sign up, as well as signing up and getting that information in your inbox. You're also going to be entering into the drawing at the end of the night. We're going to give away 6 of these do-it-yourself mushroom kits. So we're going to give some of these away at the end of the evening, so be sure to put your name and your address down there legibly so I can read it. And then we'll give those away. So I wanted to start with a brief presentation. This is a hands-on workshop, kind of. I mean, it's going to be my hands, but I did want to go a little bit into what exactly mushrooms are. Would you hit those lights, Shirley?
Where is it?
Right behind you. All right. So, as I was saying, my name is Gil Lopez. I don't know if I got to that part, but I do work for the Queens Library, as well as several other things. We do have lots of other programming here. We are funded by the Greening Western Queens Fund, which is a grant that was given out to a specific territorial region here in Queens, and we are greening the Queens Library in many ways. The grant is specifically to be used in a territory, like a region of Western Queens. Right now it encompasses Astoria, Steinway, Broadway, Sunnyside, and Woodside branch. We're expanding to the Long Island City branch and the Ravenswood Adult Learning Center in the near future. So we're really excited about those expansions. We're also bringing on another part-time employee that's going to be a counterpart of myself. So more programming like this, more children's programming during the day, more compost collections, lots of fun stuff. So tonight we're going to be talking about growing your own edible mushrooms. I wanted to just stress that fungi are completely different they're not plants, they're not animals, they're in a completely different kingdom of species. So just to give a broad overview, I'm not going to go into depth of any of these concepts because we've got some hands-on work to do. But they're not plants, they're not animals, they're not bacteria. As a matter of fact, they compete with bacteria in the forests and in the soil for nutrients. So the fungi kingdom includes, I'm sorry, you can't see. It includes, mushrooms like you and I imagine when you come to an edible mushroom workshop like this. It also includes yeast, bread molds, lichen, and a host of other kind of living organisms that some people don't necessarily attribute to the fungi family. Today we're going to be focusing on mushrooms in particular. There are a few different types of mushrooms. There's mycorrhizal, parasitic, and saprophytic mushrooms. We're going to go a little bit more into depth on each of these. Basically, mycorrhizals work in conjunction with a symbiotic relationship with the roots of plants. Parasitic mushrooms live on living organisms, whether it be plant or animal or bacteria. And saprophytic they're decomposers, so they decompose dead material. So first, the mycorrhiza, it's a symbiotic association between fungus and the roots of a vascular plant. Generally, this is a mutualistic relationship, but can occasionally become a weak pathogenic relationship. So a lot of gardeners, is anyone here a gardener? Do you use mycorrhizal inoculants when you germinate your seeds? I would highly suggest checking that out. Mycorrhizal inoculant can be purchased. It's a very fine powder, and you can just dip your seeds in it whenever you're doing your spring planting. The mycorrhizae grow along with the plant roots, and they help the roots. You can see here a plant with mycorrhizae attached to it. The mycorrhizae reaches out. It grows on the root of the plant, but it reaches out deeper. It decomposes and makes nutrients bioavailable for that plant. It brings it up to the roots of the plant, and it forms a living network in our forest. Studies have been shown that these types of mushrooms can communicate between the trees on the outside of a forest to trees on the inside of a canopied forest and displace nutrients accordingly to plants that need nutrients that aren't getting the sun, and the plants on the outside that are getting more than they need. So these are really beneficial and necessary for development in the forest. Parasitic fungi are members of the kingdom which thrive by latching on to other organisms and taking nutrients from them. They're a type of symbiotic fungi, but they are not mutualistic because they do not lend themselves to giving anything back to their host in return for sustenance. So I have an image here of a little ant that has been consumed by a parasitic mushroom, and then this is a living tree that parasitic mushrooms are actually growing on. They're getting into the living plant tissues of the tree, which is a lot different than what we're going to talk about next, the saprophytic mushrooms, which decompose the dead tissues of the woody plants. So the animal parasite is kind of interesting. It's also known as the zombie fungi because it attacks the ant or spider or wasps or whatever it is, and it gets into their system. It starts taking over the interior of their system, and it gets into their brain, and it tells the animal to go to, and it kind of controls the animal, makes it go to kind of a high point where it can grow and form a mushroom and sporulate and cast its seed, its spores through, and it can be cast as far as possible through the wind when it's at that high point. Carbonopsis? I'm not sure.
At what point does it parasitize the bug?
At what point?
Yeah, like when they're larval or before that? When does it get in there?
That's a good question. I hope that all this opens you up to a whole new world of discovery. There are books available here at the library. I'm going to just repeat that I am not a specialist of this. I'm just kind of trying to bring ideas and shed light on things that some people might think is a hard topic to understand. It just takes a little bit of research, a little bit of discovery, a little bit of doing, and we can tackle these things. Yes, sir.
There's an article in newscientist.com which says that 99% of those pictures are upside down. The picture on the left.
I wouldn't be surprised.
Yeah, the underside of the leaf, because either the spores will fall down onto other ants. You were asking about when it's affected. The other ants touch the spores, they become the zombies again.
Okay.
There you go.
So when they're adults.
Yep.
Okay.
So the last one that we're going to talk about, and then we're going to kind of do a little bit more talk about mushrooms, and we're going to jump into things. The saprophytic fungi, they feed on dead plant material and animal remains. Many of these are extremely beneficial, breaking down the organic matter into humus, minerals, and nutrients that can be utilized by plants. Without these fungi, we would also disappear under a mountain of unrotted dead leaves and logs. So these are the primary, secondary, and tertiary decomposers of a forest on the forest floor. They work alongside worms and other insects. They work with fungus, bacteria, and molds to decompose everything that falls on the forest floor, all the leaves, the branches, the dead animals, the dead insects, and they turn all that stuff back into nutrient-rich soils They help break down the larger chunks of wood. They get into fallen logs. A worm can't just like attack a log. It needs some intermediary before a worm can start breaking things down. Same thing with like a roly-poly or a pill bug. They need these mushrooms to start breaking things down at a molecular level to where it's accessible to other organisms to break down. So just basic information about a mushroom. This is a mushroom. The bottom of the mushroom has the mycelium, which is the part that we're going to be working with today. There's the stem, which is composed of it's an outer sheath, and it has a bunch of mycelium that go up into the cap. So the cap is on top. The whole thing that is above ground is the fruiting body. Underneath the cap, you have the gills, and that's what sporulates it. It releases the spore to be taken through the wind. There's other ways that mushrooms release spore other than through gills, but we're going to be focusing on this way right now. The mushroom is actually the smaller part. It's the fruiting body of a larger organism. So in this slide, they call the mycelium hyphae. But starting over here at the fruiting body, which is the part that's above ground that most people are used to seeing, that gets fertilized by dropping the spores. Those are get through the diploid and haploid stage. That's a mono and multiple cell stages. When the nuclei of the spore kind of start to come out, it creates this hyphae or mycelium. I'm going to use those terms interchangeably. But when two mycelium from different parent spores come together, then they can actually exchange DNA and grow another fruiting body. So that's how the mushroom multiply. And let's go to the next slide here. Yep, so that's it. So yeah, we're going to go into the actual workshop portion of this now. Good. About 15 minutes. I'm going to turn the lights on real quick. Thank you. So today we're going to be working with some myceliated, I'm going to rip this for you guys, some myceliated sawdust, which I bought from Fungi Perfecti, which is the name of a company by a very prominent mycologist, Paul Stamets. He's written many books about mushrooms, about propagating mushrooms and growing mushrooms at home and in your garden, in the lab. Oh, wow. So these usually have a little booklet, but it's not in this one this time. That's usually my cheat sheet. Uh-oh. We'll see how well I know my stuff. I'm not going to open this all the way, but this is sawdust. And all the white stuff on it is the mycelium, which, as I was telling you, is the kind of infant phase of the mushroom. So all of this can basically be put onto another growing medium to multiply and grow into a larger body of mycelium. And then the more material that the mycelium has to munch on, to eat, to decompose, the more mushrooms you can start harvesting. Now you can use this and you can inoculate. You can insert this into something that is 5 times as bulky as this. This is about a gallon-sized bag. So we can inoculate another 5 gallons of material. Today, we're going to be starting things in this milk crate. So it's important to, when you get material that you're going to be using to inoculate, you have to disinfect it. Well, you don't have to, but it really helps the process if you disinfect it, if you kill off all the other bacteria, any other fungi that may have gotten into the material, any other bacteria, molds, anything else that might be in there that would compete with. This is oyster mushroom. If I had my cheat sheet, I'd tell you the scientific name. It's Ostrum pelastrosum or something like that. I'm sorry that I'm halfway without my material. It's really unfortunate. But there's a few different ways to disinfect your growing medium. Today we're going to be using wood chips and coffee grinds as our growing medium. So coffee grinds are great. Like I was saying, this little guy right here This was created by Back to the Roots company in Oakland, California. They use coffee grind pretty much exclusively. They collect it, they put it, they pack it, they get it to a certain moisture temperature. They inoculate it with oyster mushrooms again. Oyster mushrooms are pretty much the powerhouses of the mushroom family that a lot of people like to use. They're really easy to work with, and they're kind of like the go-to for beginners. So coffee grinds are great for it. One reason the coffee grinds are great is because when you brew coffee, you put really hot water through the grinds, and that disinfects everything. So in that process, you're basically disinfecting the whole batch of coffee. So once it's disinfected, you just need to gather it. Don't give it a whole lot of time for any other bacteria or mold or anything else to grow on the coffee grinds. They package it up in a plastic bag, kind of airtight, so that when you get it, you can cut a little X in it, and you can soak it overnight in water, and then you can mist it every day, and then you start growing mushrooms out of the front. You take this little panel off the box. What we're going to do is we're going to put wood chips and coffee grinds in here. Now, for our wood chips, what you can do is you can boil them and keep them at, I believe it's 160 degrees for about 30 minutes. And that sterilizes the batch of wood chips. You can also, so not necessarily wood chips, but you can also use other things like straw or sawdust. And if you were to use straw or sawdust, you could, along with boiling, you could also use hydrogen peroxide. You could dilute your hydrogen peroxide to 0.3%. Most hydrogen peroxide that you get at the drugstore is 3%. If you're working with culinary or agricultural hydrogen peroxide, it's usually 30%. You want to dilute it to 3%, and you can soak straw, or if you wanted to use coffee grinds in like a burlap sack, you could soak your burlap sack in the peroxide for 24 hours or up to a week, and that would get rid of any bacteria or anything that could compete with your mushroom spawn. What I've done is I've put the sawdust, the mulch, the wood chips in this bucket, and I filled it up with water. And what that did is it created an anaerobic atmosphere where only anaerobic bacteria could form. So we don't want the anaerobic bacteria necessarily to be on our wood chips. But the thing about anaerobic bacteria is whenever it's exposed to air, it dies. Just like aerobic bacteria is dead in here. I've had these wood chips in this water for about a week and a half. A week is prescribed. Anything longer than that is good. You know, the anaerobic bacteria, whenever you expose it to air, it dies, and it actually becomes food for the mushrooms to grow on. So you're just kind of giving the mushrooms even more added food to eat. So what I'm going to do.
So what kind of wood chips can you use? Like, can you use
that's a good question.
From pine trees where there's a lot of tar and pitches in it, or
yeah.
can you use oak where there's a lot of tannins in it? You know, where can you?
Generally, you want to do the research on what sort of mushroom that you want to grow. For example, I've been told that shiitake is Japanese for oak.
Right. And what they talk about is you get an oak log.
Yeah, log.
Yeah.
And you drill 3/8 holes in the log.
Yeah.
And
So
then you
that's
get the
the log
spore
inoculation.
plugs, and you insert the shiitake spore plugs into the log. And it takes about 2 years to colonize the log.
Yeah, that's a little bit different way than what we're doing it tonight. But that's definitely one way to do it. So different mushrooms grow on
I
different
got 50 acres upstate, so.
I haven't heard of a whole lot of mushrooms that really enjoy sappy wood. So in general, you want a hardwood with low sap content.
But would like sugar maple be better? It has more food for them. I'm understanding that wood chips already pre-broken down is going to take a lot less than 2 years to colonize because
Right.
it's not the density that.
Yes. That's correct. And another factor that you want to think about is how long the tree has been dead. You want to find a tree and get wood chips from a tree that's been dead for more than 2 weeks, but less than 2 months. Because more than in 2 weeks, you still have the antibodies and the tannins in the wood that don't allow saprophytic mushrooms to grow. Parasitic mushrooms will grow, but oyster mushrooms are not parasitic. So you want to give it time for the tannins to get out of the tree, for the tree's cells to stop producing tannins, and for them to just be gone. But you also want to do it before 2 months, because after that you have other fungus that start to come in and colonize and break everything down, and then you're having partially broken down wood. And you want those nutrients to feed your mushrooms, not to feed some other and then kill that mushroom strain and then start doing it again. So it's best, that's the little windows, 2 weeks to two months, that you want to try and get your wood. So what I'm going to do is I'm going to put this the milk crate right in my, this is just a big tote that I have. We actually use these to collect compost. I cleaned it out, but it doesn't really matter if it's clean or not because we're just dumping water in it.
Where do you get those totes from? I think especially sturdy.
They are. They're Rubbermaid Brute industrial totes. I got those online. I think you can get them at like any hardware store pretty much, but you can get a better deal online in general. So I'm going to try not to make too much of a mess when I pour this in here. The rest of the library staff wouldn't be happy. Actually, they're going to have to clean it out.
What kind of wood chips are these?
This is a mix of wood chips. I came across a contractor that had a contract to remove trees from the cemetery in Queens, and he dumped a 35-yard load at my garden in Long Island City.
35 yards.
You don't know what type.
Yeah, you guys probably don't need 35 yards.
Oh, we could collect it when it's clear enough.
We could. Doing things collectively is good.
So you don't know what type or?
They're primarily hardwoods. I don't know exactly what type they were, but I did talk to the contractor, and he did assure me that they were primarily hardwoods. So I'm going to put some gloves on. This is not a clean room in a lab, but I'm going to try a little bit to keep things a little clean. It's not going to be the end of the world if we get something else on it. We are going to put a lot of this mushrooms grain or wood shaping spawn onto this. So I'm going to spread this out. Let it all get a little bit of exposure to the air. I'll put this down so you guys can see. While that probably needs like at least 5 minutes. But while we're waiting for that, I'm going to put this cardboard and I'm going to make a little kind of tray so that everything doesn't just fall to the bottom whenever I start doing that. And I tried to do it real quick before the class started, but I made it too small. So I'd love to field any questions while I try to get this going, because I know that there's a lot of things that I didn't really touch on yet that I'm sure you might be interested in. Anybody have any questions?
What about the non-edible mushrooms? They grow faster competitive?
Yeah, it'll take.
Could you repeat the question for those listening?
The non-edible mushrooms.
Oh, the non-edible mushrooms.
Yes. They can amplify themselves.
Non-edible. On this? Like, could you just like let that grow mushrooms? Yeah, I mean, mushroom spores are all over. They're in the air pretty much everywhere. And for a spore to kind of take hold, of a certain location is kind of hit or miss, you know. It's better if it falls on something that is very hospitable for its growth. And that's why we're going through all this, what may seem like unnecessary steps to try and give it a clean environment for it to kind of thrive.
So what if you fail at that? What if mold grows? What happens?
What if mold grows?
Yeah, is it done? Is it over? Or
I mean, mold and mushrooms are always competing in nature.
Yeah.
It doesn't necessarily mean that it's over, but it might take a little bit longer. There might not be as much food for the mushrooms that you want to grow to eat. But I think that if you give it time, you can see. You know, I mean,
wait it out.
yeah, I would wait it out. I mean, I wouldn't just toss it out. I would make sure that it's not moldy inside my apartment. You know, you don't want mold inside your apartment because that can lead to, you know, illness of some sort. I'm sure. I'm not a doctor.
Black mold is a very
Black
bad
mold
thing.
is terrible. Yeah, so that fits nice. There we go. Ta-da! All right. So the other thing that we're going to do is I'm going to get some coffee grounds. So I just called Starbucks up earlier today and I said, hey, will you save your coffee grinds for me for the next few hours? I'll be there at 5:30 to pick them up. And I went at 5:30 and picked them up, and here they are. So this is another thing that you can kind of get for free, or at least one thing that you can get for free. And they'll grow on the coffee grinds, you know, no problem. You don't have to have all these wood chips. The wood chips are a great bulking agent, and it's a little something different. Yes.
Do they like the acidity?
Yeah. Yeah, they love the acidity. They're really they're fine with it. Like I was saying, that back to the roots box. It's just coffee grinds inoculated with mushroom spawn, and they'll be fine growing in that. They don't need a whole lot other than just the coffee grinds. But I like to add something else just for demonstration's sake. And if we had just coffee grinds, it would all fall out of the sides of the box. I mean, we'd have to do our cardboard box a little bit different. And ideally, in the ideal world, if I had more time to plan for these programs, I would have soaked the cardboard box for an hour or two in the hydrogen peroxide solution, and it would be super moist as well. But I think we'll be okay.
Now, what about the black print that's on the cardboard?
It's fine. It'll be fine. Most of the inks that are used nowadays are soy-based, and even at that, mushrooms have been shown to be phytoremediator or microremediators.
Bioremediators?
Yeah. So bioremediation is a term used to talk about cleaning toxins from the soil using natural processes. Phytoremediation is using plants. Microremediation is using mushrooms. Yes.
Yeah, but doesn't it concentrate whatever toxins into the mushroom itself?
There's a lot of different ways that bio and microremediation work. Bioaccumulation primarily happens in plants, which are uptaking heavy metals. Mushrooms don't really bioremediate heavy metals. They primarily remediate volatile organic compounds and other compounds. And what they do is it's not accumulation, it's a breaking up and a molecular restructuring of those compounds.
So you don't have to worry, let's say the wood chips is full of heavy metals, it won't go into the mushrooms because they pretty much leave it alone.
My understanding is that it's not going to take up the heavy metals. Now there's other things, if you're growing like collard greens in soil that has heavy metals, those will take up the heavy metals. But this is a little bit different. This is a lot different process. So it's not something that you need to worry about. So
Can you say how much this costs?
this cost me about $18, and then about $20-something with shipping and handling.
Okay.
I got it on sale because I got multiple ones, and there was like 20% off if you did that. So I actually got it for $15. So, but it cost about the website's called Fungi Perfecti. I think it's just fungi.com. That's,
I know there's a company, I get the catalog every year, all they sell is mushrooms, and it's in Washington State someplace.
yeah, they're based in Washington State, and Paul Stamets is my college's up there. He teaches classes. You could probably find out a lot more information than I'm telling you.
Give a ratio, coffee grounds to blue chips, or anything?
Yeah, 5 to 1. So you can inoculate 5 times the mass. Like basically, this is 1 gallon. You can add 5 gallons more to this and end up with 6 gallons of material.
No, the coffee
To chips ratio.
Oh, that's completely arbitrary. It can be 100% coffee or 100% chips.
Okay.
I actually didn't use chips a few weeks ago. I used newspaper, which is just cellulose, and I boiled it for an hour. I kept it at temperature for 45 minutes, and it was really hot, and then we put it in there, and I mixed layers of newspaper pulp and coffee grinds and myceliated sawdust, and we did that several times, and it's already, that was that was 3 weeks ago, I think, and it's already, you can see the mycelium growing on the sides. I should have brought a picture of that, but that's pretty interesting. So I'm going to go ahead and open this up, and how am I doing on time? Yeah, we got about 20 minutes left, so we should go ahead and get this rolling. So it's good to break it up kind of in the bag. The more that it kind of like sticks together, the more it gives a way of how strong the mycelium is in there and how well it's doing. So all this it's pretty good and clumped up. All right, so I'm gonna put a base layer of wood chips down here in the bottom, and I'm just gonna put like a couple inches. Closer to my box. And this also has, the filters. The filters are fine, it's just more cellulose. It's basically a wood product. These are probably unbleached and all the good stuff that Starbucks is all about, whatever.
Do you want to do it in a dark area or well lighted?
Well, I mean, the mushrooms, typically, the mycelium grows underground, and they need darkness. So we're going to talk about that a little bit more. But essentially, you want to, after you create this, you're going to want to put it in a dark area for a little while, and I'm going to want to put this. Actually, that's one thing I forgot to do. But I need to get like a translucent bag and put this around it and put some holes at the bottom. Mushrooms breathe in oxygen, and breathe out carbon dioxide, just like we do. So you don't want the carbon dioxide accumulating where the mushrooms are trying to start growing. So I just, I put some coffee grinds on top of all the wood chips, and I'm just going to grab some of this, guys, and spread that around on top as well.
If you open a bag, do you have to use the whole bag?
Yes. It's going to, I just stuck my hands in there with coffee grinds in it. It's been contaminated now. So it definitely needs to be used whenever you open it. If you have a clean room, if you're in a lab and you have a hood and you know what you're doing, and I know people that know what they're doing, they say that they can open it and that it'll be fine in another couple of weeks or whatever, but not only do I not know what I'm doing, we're not in the clean room, and there's no hood right here. So this bag needs to be used. I'm actually going to leave a little bit, and I'm going to do kind of a mini workshop with some kids in here tomorrow. We're gonna make a bunker bag, which is just I'm going to disinfect a coffee bag and with the hydrogen peroxide, and I'm gonna disinfect that for 24 hours, and I'm gonna use that bag. I'm gonna fill it mostly with coffee, and we're gonna just put the rest of whatever's left in this bag at the end of the night. Hopefully it'll last for one day. We'll see. This is not scientific. This is not science that we're doing right here. This is kind of a community project.
Advisable to cover the nose, mouth, do that.
You could go all the way. Yeah, you could put on a whole clean suit if you wanted to. Yeah, I mean it's not going to get you sick if that's what you're wondering. But yeah, I mean there's varying degrees of cleanliness, I guess you could say.
You don't have to worry about inhaling the spores though. That's what you're worried about. This is the mycelium.
Right. There's no spores involved right now. This is just the mycelium. It hasn't grown into the mushroom. When it grows into the mushroom, you need to harvest it before it starts to sporulate inside too much. Some people do have an allergic reaction to mushroom spores. And the more that people are in contact with mushroom spores, the more likely you are to develop an allergic reaction to the mushroom spores. So that's a good point. Thank you for bringing that up. So I'm just going to keep on layering this very non-scientific manner.
Jill, when it comes to harvesting, I'm noticing in your picture on the left-hand side that there's a lot of fruiting bodies on the outside of the container.
Yes.
But there's a large mass that's inside the container that is not fruiting. Can you get more yield by taking it out of the container and rolling it into cookie doughs or cookie tins or something like that to get more surface area?
No, not really. I mean, the mushroom is the fruiting body of a very large organism that usually grows inside a dead log or underground. It needs the mass inside to grow. I hate comparing it like this because it's not a plant. But if you think of it like an apple tree has leaves and limbs and trunk and roots, but all we really pick and eat is the apple. So, there's not a good way to I guess you could exfoliate an apple tree and get more apples from it, but this is a pretty good method for maximizing the surface area and the volume to get it to fruit as much as possible.
Well, so eventually the mycelium will eat the material. How do you keep feeding it after if it's all compacted in the crate?
How many harvests are you going to get out of this?
Yeah, so there's a first harvest, and it's usually the biggest harvest, and it's usually, depending on the temperature, you need to keep the temperature between 55 and 75 degrees, and it will do very well at the higher end of that range. And and you usually get like a second and sometimes a third flush, depending on how nutritious the medium is, and all the variables around, like how much moisture. You need to keep it moist. It doesn't like to be wet, but it needs to be moist, very humid atmosphere, and the temperature. Yes?
Could you keep these in a series of crates, like outside instead of inside?
You could. They're not going to do anything right now because it's too cold.
Right.
But yes,
You got into
definitely.
these temperatures.
Yes, definitely. That's something that is done.
And then a regular rain would harm it.
Regular rain is okay. You don't want to let it get too much rain. If it's you want to pretty much cover them up and let it collect water around them and keep temperatures high so you have a high humidity around them. And if they start to dry out, you want to mist them very well and keep them moist, but you don't want to soak them and drench them necessarily.
And, as you're getting to the end of your second and or third flush, would there be any benefit to taking the growing medium, cutting it to 4, and starting all over?
That I would suggest that. I don't know, that's just kind of multiplying your spawn. That's giving it, fresh material to eat. In this application, I don't know how it would work, but that's definitely something that Paul Stanley suggests whenever you do like a mushroom patch in your yard. Just keep on giving it fresh mulch every season and letting it eat its way through. If you don't do that and you just kind of let it run its course, afterwards you just have really great mushroom compost that you can use in your garden.
Yeah.
So I'm not going to fill. I'm going to put one more layer on this, but I'm not going to fill it up. I'm just going to fill it up to the handles, and then we'll have about 15 minutes left. This one went a lot faster than the last one did.
What do you think, Chris?
Faster.
I guess so.
instruction sheet.
Yeah, without my instruction sheet, but I did pretty good. Yeah, it was just a crush. So, just one more layer, last little bit. Wow, man, there's more coffee grinds in here.
For tomorrow.
Well, I'll get some fresh ones for tomorrow. I'll just call them in the morning and say I need some more.
And they were cooperative, Starbucks was. Sure. come
Yeah,
get our
Starbucks,
coffee sandwich.
if you call them or stop by and tell them that you want them to save that's a policy that they have.
Yeah.
They'll save
Oh,
save
really?
the grinds for
They
you.
actually have a policy?
They have a policy, yeah.
We've stopped at Starbucks before. Some of them have like
Big silver
have
bag.
a whole
Some of them have a little
packages
thing.
of them already
Yeah,
yeah, but
they
pre-made,
put
made
them
for your garden.
in the, by the trash
More people gonna come in
thing at the
Oh.
front, where you can just like
Because
take
then it costs
take
a
a
little
bag.
business.
Yeah.
If you're
But
taking your coffee grounds.
No, nobody really does
They don't
that.
pay
I
for
haven't
the coffee,
seen
they
that
don't
that
have to
around.
pay.
I haven't seen it around.
But you can call them
It's
and
a
they'll
win-win.
gladly
Right.
save
All the organic farmers and mushroom growers, and them.
save it for you.
Bill, if you're using just coffee grinds, I mean, it might, for some of us, it'll be easier to source the coffee grinds instead of the wood chips. Is that-
This is definitely true, and that's a big reason that I use the coffee grinds in these demonstrations. I did want to mention, kind of as an aside, that I, not involved with the Queens Library, will be purchasing more of the grain spawn. So I'm going to be making an order from Fungi Perfecti. If anybody is interested in ordering anything, we can, split shipping.
Do bulk orders and split shipping.
Yeah.
Very cool.
If anybody's interested in that, just let me know after the workshop.
To that place on this line, it's the hydroponic store. They get a lot of really great stuff in there. They get home food kits. They got wine
If you
making
want to
kits,
do it
they got
commercially,
hydroponic kits.
you're better off not buying the
That's good
spore
fungi
from
and bacteria.
Fungi Perfecta. You're better off multiplying it in a lab and, becoming really knowledgeable on the topic and knowing what you're doing and investing some money in lab equipment and a clean room and all that good stuff. But it can be a highly profitable business if you
have I was thinking about the backyard kind of thing, like I have a big backyard, why not to use it instead of just giving up?
Yeah, that's a wonderful idea. I would completely advocate for that.
I'm wondering if somebody else knew about that.
Yeah. So that's pretty much it. What I'm gonna do is I'm gonna get another bag. I might even just use this one.
It's on the north side.
Why not? Reduce, reuse, and recycle, right?
Right. You're not worried about that being sterilized?
No, I mean it's a trash bag. They probably got a brand new fresh trash bag at
It's out in
2 o'clock or whenever I called them, and they double bagged it.
Yeah, they did.
And then they just put the coffee grinds in it.
The last time you did this, where did the mushrooms come out? All around it, or just at the top, or?
Well, the last one that I did was only like 2 or 3 weeks ago. So actually the mycelium is just starting to run. You can see it coming onto the cardboard in this like dendritic pattern. It's pretty cool. And then on the newspaper and the coffee grounds on top, it's starting to spread out and really kind of take over the whole thing. So no mushrooms have grown yet. This is going to take 2 to 3 months, depending on the temperature, to actually start fruiting. And then after that, it'll fruit for maybe like 10 days to 2 or 3 weeks. And it'll go kind of into a hibernation. I forgot the technical term for it, but it'll kind of retreat. And then it'll start to, and then 2 to 3 months after that, it'll start growing again. Yes.
Two really quick questions. One is, so if you can get it at a higher temperature, like closer to 75, you can take a shorter period of time?
Well, it needs to be at the higher temperatures during its hibernation, and then, while the mycelium is kind of taking over this whole mass of material, it needs to be at those higher temperatures, high humidity, relative low light, and then whenever after that 2 to 3 months, when you start to see, I'm forgetting the name of it. There's a name for the little baby mushrooms when they start to come up, and they're just so cute when they're little. they start to have like these lumpy little things that start growing, and at that point you want to expose them not to direct sunlight, but you want to take the bag off kind of completely, maybe leave it down on the bottom just so that it can have the moisture and the humidity retention down in the bottom, and you want to keep the temperature at a more moderate range. It doesn't have to be quite as warm. And then, yeah, not direct sunlight. It could even be fluorescent light for 8 hours a day, or it can be right beside your window for during daylight hours, unless you live in a really bad apartment that doesn't have really good lighting. Like I know a lot of people that don't have good lighting, so they would have to find another place, or rely completely on, it can be direct, like phosphorescent or artificial light, but not not direct sunlight. It doesn't want UV radiation hitting it directly.
Can you keep it under a tree?
Yeah. Yeah.
In a shady area?
In a shady area, yeah, that would be great actually. And anecdotal studies have shown that if it's actually in contact with the native soil, then it does better. Now, I don't know why it's antithetical to meat, because there's competing bacteria and everything else in the soil, but
It knows its home, it's not in an artificial environment.
yeah, right. So we have about 10 minutes left. I'm going to go ahead and, here, I'm gonna wrap this up. I'm going to cut a couple slices in the bottom of this. I'm going to tie up the top very loosely. I'm going to double knot it, but I'm going to leave it pretty loose. And I'm going to make sure that it's open. There needs to be, air exchange. It can't be, this is not an anaerobic process. It needs to have air exchange.
And how much moisture?
How much? It needs to be like kind of moist to the touch. And you can touch it. I mean, you need to wash your hands, or desanitize your hands, use hand sanitizer or whatever whenever you mess with this. But it needs to be moist to the touch. I'm just going to cut a couple slits around the bottom of this. Nothing crazy.
So you're kind of anecdotally experimenting with these different mediums in there? And these
Well, this is totally legit. Last time I did this with
the
newspaper, that's not something that Paul Stamets really talks about, but I do know that there is a group in the Bronx that are growing mushrooms on newspaper. So it's done. Yes?
Yeah, so that's the method in a crate. So what if you just put all that stuff in there? Can you don't need a plastic bag, can you just leave the top open just a little bit, like a crack, to get the air in?
Where, in what?
In like something like that.
In this?
Yeah.
Actually, Paul Stamets does recommend, he doesn't recommend a crate. He recommends a 5-gallon bucket. And what he recommends is fill it up And whatever the top of your